Hello, everyone, and welcome to the RDCA-DAP Prototype 3 presentation. This is related to our effort in polycystic kidney disease modeling. A little bit about myself. My name is Jackson Burton. I currently lead the Quantitative Medicine program at c-path, and what we really focus on is the development of a lot of quantitative solutions across a lot of different disease areas that are really focused on addressing key drug development questions. So this one here is kind of an example of similar things we do across the board. And so I'm going to go a little bit into the details about what we've been doing here with PKD. So PKD is really an unmet medical need. There is currently only one drug that slows the progression to end-stage renal disease, which has been approved by the FDA. So what we want to do is understand the vision of RDCA-DAP in the context of PKD, which really is about how can we leverage patient-level data to best enhance decision-making for clinical trial design. So the goal that we have within this pilot project is really to use model-informed drug development approaches, which I'll talk about in a little bit, to really take the patient-level data and develop quantitative tools that will be predictive of end-stage renal disease. In the context here, we're going to focus on what data we have, which has estimated glomerular filtration rate, or eGFR, and total kidney volume as longitudinal markers to really inform when we think that end-stage renal disease is going to occur. So conceptually, the way we actually do this is really an intersection of three concepts, all of which lend to the idea of model-informed drug development. So of these three, the most important that we have to focus on is data. The data sources that we get have to come in and really be used as a basis to inform all of our quantitative efforts. So within the Data Collaboration Center at c-path, in the context of RDCA, we're able to integrate and aggregate data from multiple studies and use that as the basis to generate the analysis subsets needed to build models. And that's where the quantitative medicine program comes in that I lead, for which we use a number of different quantitative tools to build these models. However, we cannot do this in isolation. We have to make sure that in the collaborative sense within RDCA, we have the scientific expertise, subject matter knowledge experts, and external stakeholders, especially regulators, who can help along the way inform the process so that it ensures that it is clinically useful for sponsors to eventually adopt. So the intersection of these three concepts is really where MIDD flows from, and it's the basis for how we're going to be doing this work. So where we always have to start with is the data. So a quick summary of the data sources that we have and what we're going to be using. 5 observational studies were integrated and standardized according to CDISC terminology and structure, which is called SDTM, or the Study Data Tabulation Model. By taking this approach, we were able to put together these 5 studies and from there have a harmonized interoperable set of data for which we could extract an analysis subset out of. This subset contained 587 patients, just shy of 2,400 total longitudinal records. The key thing here is that the longitudinal measures of total kidney volume and estimated glomerular filtration rate are captured in addition to the end-stage renal disease events. So this serves as the basis for which we're actually going to carry out the modeling. Before we actually tackle any modeling, we always have to build intuition about what the data looks like through both exploration and visualization. So some of the key variables here that I'm just highlighting, of course, are eGFR and TKV that I've just mentioned. And we always like to look at these things from both a baseline and longitudinal perspective to really get a sense as what would be the most appropriate modeling methodologies. So here we're just showing some visualizations to get an understanding of how things look initially and how they change over time for these 2 longitudinal markers. What's important here is that these markers we know are established in the literature as being linked towards end-stage renal disease. And within the pathological considerations of polycystic kidney disease, we know especially that total kidney volume, which was previously qualified as a reasonably likely surrogate marker, connect very closely to end-stage renal disease. Hence, this is why we are focusing on these longitudinal markers. In addition to that, beyond these markers, we also have to consider the different patient characteristics that could influence the risk to end-stage renal disease. So examples shown here is with a chronic kidney disease staging criteria for which different stages actually have very different profiles towards end-stage renal disease, where each curve there represents a different timing towards end-stage renal disease within those groups. And so this process is done across all the different patient features of interest, all the different longitudinal information of interest, and really gives us a good intuitive sense of how we can start executing on model building. So once that's done, we can really move into the quantitative landscape. I realize not everyone really enjoys looking at equations. I personally do, but I'm putting these up here really for the purpose of tying them to interpretable aspects of what we're doing in this clinical sense. So these models here have a lot of complexity to them, but really they just boil down to things that we can understand. For example, within the data, because there's a lot of variability, we need to quantify that variability, especially between patients, and that's really necessary for understanding how we're going to design clinical trials in terms of how to power the studies. Additionally, as I mentioned before, those patient features that we know are likely tied to end-stage renal disease, well, we want to capture those as well to understand how they can be used to improve the accuracy to that timing. Further, we also want to take into account the longitudinal markers, which is shown in this bottom equation here. And those are then used to really inform the timing of end-stage renal disease, which of course is critical to the clinical trial design itself. And ultimately, this is wrapped up in this final notation here at the bottom, this H of T, which is really the end of where the end of where the modeling goes, which is how we actually predict the timing to end-stage renal disease. And that represents, for the most part, the clinical trial endpoint that sponsors are going to be interested in powering their studies off of. So the totality of all this, and there's far more equations that are shown here that go on behind the scenes, really represent how we connect both the data and the quantitation to be interpretable. So once we actually do that and we carry out the model building process, we actually get numbers, right? We always get numbers out from models, and the question is, well, how do we interpret them? And because we have used rich patient-level data sources, we create these models in identifiable ways, meaning interpretable ways. We actually get out numerical values for these parameters that actually connect to clinically relevant phenomena. For example, some of these different parameters tell us the effect of, say, how that chronic kidney disease staging affects the timing to end-stage renal disease. Similar things like baseline age can be used to link to the change of end-stage renal disease, and also how important total kidney volume and eGFR are in their effect on end-stage renal disease. So in doing this, we actually get these interpretable parameters that are going to make their way into how we build a tool for informing decision-making for PKD study design. Once we actually do this, though, we actually have to do our due diligence of making sure that the models are properly tested. So there's a stage of the model building process where we actually evaluate them and we perform diagnostics and performance testing. So on the left here, we actually show these plots that are basically sanity checks to make sure what model we've actually built lines up with the data, that what we predict and what is observed actually correlates with each other, for which this model does show a very high degree of predictive accuracy. Further, we also want to understand how that predictive accuracy is connected to the particular context of the disease, in this case, end-stage renal disease. So in this plot here in the lower right, what we're showing is how the model actually predicts the timing of end-stage renal disease based on the observed data, which is shown in the pink here. And so what we see is a very high degree of correlation between what the model predicts and what the data shows, giving us confidence that when we actually do, say, a simulation, that we can have confidence that this model is going to be accurately reflecting what we see in actual patient populations. So when we do all this, and we've done our, the effort to actually, look at the data, curate it, standardize it, integrate it, build the models, interpret the parameters, do the diagnostics, we then look forward. And we want to start putting this in the context of an actual practical tool. So the developed model that I've presented so far is really intended to be used as a basis in a clinical trial simulation tool. Well, what is that? A clinical trial simulation tool essentially has three components as seen in this schematic here. It has a disease progression component, which is represented by the model I just showed you, but it also has other components that are relevant for clinical trial design, such as trial components, which is the way in which we can actually incorporate things like placebo effects or patient dropout or different trial designs as it relates to the disease progression model so that it can better inform decision-making for trial design. And then lastly, we often, when data permits, want to incorporate drug effects. And these are ways of actually simulating the trial so that we have a difference from a placebo group and a treatment group, and we can understand how those differences spread out based on these different features of the clinical trial simulation tool. So an example of a clinical trial simulation tool is shown here on the right, one that we've previously made at c-path for Alzheimer's disease. And what it allows users to do is essentially choose the different parameters of interest that they would want to design their trial. For example, what would be the inclusion or exclusion criteria for their patient population of interest? What type of enrichment strategies would they like to use? And ultimately, what duration of trial, how many patients should be included? And all of this is used in totality to really understand the power of the trial. So how likely is the trial to succeed? And I want to emphasize that we're not proposing that we or suggesting that we should remove the need to perform a well-executed trial, but this is really used as a decision-making tool prior to the design and execution of a trial and really help de-risk drug development in this context for eventually PKD. Because ultimately, at the end of the day, if a trial is going to fail, we want it to be because the drug was not efficacious, but not because the trial itself was poorly designed. So this is where we're headed as we continue to develop this work. And so in summary, I just want to wrap up and say, really what we're doing at the end of the day is taking data and fundamentally transforming it into knowledge. If you do type in Google data to knowledge transformation, you are going to get some permutation of this pyramidal schematic here. And really, at the core of it, that's what we aim to do with data. And in the context of PKD, it's really being enabled by the collaborative nature that comes with RDCA-DAP. It gives us the ability to generate these tools all the way from data acquisition to the model development and vetting that against the disease-specific expertise that comes within the collaboration. So we're really glad to have had this opportunity, and this work is going to continue to go forward, but we just want to thank RDCA-DAP and the ability to do this and look forward to continued work coming out of this. Thank you.